So without any further ado, let's go into the talks. Looking forward to these. We'll at the end of each talk, we'll just have a brief minute in case we want to do some editing afterwards. But let me invite Alistair, Director of Science for the Foundation, visiting professor in quantum biology and bioenergetics, the Research Centre for Optimal Health, University of Westminster. Alistair, you now have 6 to 8 minutes, or 5 to 6 minutes.
Okay, well, thank you, Jeffrey. Okay, hopefully most of you will kind of remember what I talked about before, but basically we're going to propose a hypothesis which kind of links everything together from a certain angle. And that's the idea that perhaps electromagnetic fields were important in shaping life right from the word go. Next slide, please. Okay, thank you, Lena. So why is understanding the origins of life important? It's always quite good to go back to the source in the beginning, and it's often been thought that if we can understand the origins of life, we may be able to answer the really hard questions like consciousness, are we alone in the universe, organisms, shape and form, and in particular, we might be able to understand a bit better about aging and regeneration. And of course, that latter point is quite important in understanding medicine and potentially how we can, for instance, reduce disease. Clearly, if we can slow the aging process or even stop it and help regeneration, we may all live quite a lot longer and therefore have less disease. Next slide. Just a brief recap. So where did life get going and how did it go and why did it start? The best theories seem to be hinging around thermodynamics and dissipation and self-organization principles, perhaps as described by Ilya Prigogine, and it may well have maybe harnessing quantum principles. The energy for life, well, there are several different areas, but the most ones that most people think it started with was photochemical or geochemical or even radiochemical. And some of you might have seen in Nature, there's been a paper now finding that bugs can live very deep and down into the mantle, living off radionucleotides, which says life can perhaps be anywhere. It seems that these principles of underlying thermodynamics seem to have organized chemicals into the ability to transfer energy. And one of those mechanisms is via redox, oxidative reduction. And then we add in other theories and mathematical formula about self-replication, informational theories, for instance, cyclobrillion. And this enables natural selection and survival of the fittest with positive feedback, which has led to self-replication and continuance of life. And it may have started, again, lots of discussions, but potentially either in the sea, meteorite craters, thermal vents, either on land or in the sea itself or in other clays or somewhere else, but somewhere else obviously is another planet. But of course, that doesn't solve the problem. Next slide. So what else could be involved? This is perhaps slightly less conventional, but are these areas important in the beginnings of life? Well, obviously photons is one, but other areas are gravitational fields, moving gravitational fields. So were moons important in starting life? Magnetic fields from the core of the planet, stuff moving, for instance, tectonic plates or even just rocks sliding against each other. And lastly, of course, electromagnetic fields. Next slide. Thank you. So the basis of our hypothesis, we call it the ethereal skeleton as a bioelectric template model, is that at the beginning of life, there is clearly energy gradients. And one of the potential sites of life beginning was actually in thermal vents. And this is where there is a big potential of energy where enabling the flow of ions. And of course, when an ion moves, because it's charged, it creates an electric field. And if we move forward, it's possible that these fields themselves can contain information. And then if you start to go into complexity, and this is the ideas of Mike Levine, that of course you can start to develop much more intelligence by cooperation. So this really pushes cooperation as being critical in evolution of higher life forms. And today, of course, we now see this idea that Mike Levine's putting forward that morphogenic fields are important not only in the shape of organisms, but also in regeneration. Next slide, please. So practical biology. One way of looking at this is to think, well, if these ions have been important right from the word go, could they have been the condensation point for the formation and crystallization of proteins or amino acids to begin with, and which led to like a condensed kind of molecule? And clearly, one of the ones which is very important is the ATP synthase, as Wayne will talk about, has talked about. And it seems that this is one of the very earliest channels to form because it captured the energy in the gradient. And it now seems that this ATPase and certainly VATPase may be very critical in the formation of morphogenetic fields, which is a new paper from the Levine's group. Bottom line, if we can couple ion flow and energy, and thus potentially EM fields, we have perhaps an early biological prerogative. And here I'll just show very quickly at the bottom is potential for how we can integrate quantum effects into this. And it's thought that in the walls of geothermal vents, there are compounds like iron sulfide of iron nickel sulfide, which have very interesting semiconductor properties. And this enabled the flow of electrons, which could be through tunneling, to start as a form of the basis for like an electromagnetic field, which allowed the formation and condensation of various organic molecules. And then next slide, please. Another aspect to this is which is perhaps less looked at in biology, of course, and certainly sometimes in physics, of course, is that all cells seem to have this rather peculiar property of uncoupling, and that is where they uncouple the energy in the proton gradient, for instance, to produce heat. But of course, when you look at this from the dissipation point of view, this enables a constant flow of ions. And in modern life, this clearly is important, for instance, in controlling redox and reactive oxygen species. It also generates heat, but it's also very good in communicating information. And what we do know is from some experiments that if you uncouple very gently, you can increase lifespan, suggesting that dissipation is quite an important mechanism in enabling structures, dissipative structures, to survive. Okay, next slide, please. Practical. Okay, so of course, from our perspective, because we're very much biologists, one of the areas we've always been interested in mitochondria. And of course, the mitochondria ancestor were prokaryotes. And of course, this is where a lot of the uncoupling seems to go on. And mitochondria produce very, very powerful electric fields. And we know that the production of energy and electron transport is coupled to the production of reactive oxygen species. And the interesting thing about that is, of course, that is also coupled, we think, to the generation of biophotons. And we also know that, of course, that biophotons can affect the electron transport chain. So you kind of got a very nice feedback mechanism there, which potentially could exist to enable some kind of homeostasis. Then if you link that into other ideas in the cell, like microtubules and bioelectric fields, you have a series of nested coupling going on, which potentially could involve quantum effects. Next slide, please. Practical medical applications. We've always been interested in natural products, but what is thought is that many natural products in plants certainly evolved to dissipate the solar potential, i.e. their sunscreens. And of course, what we now know is a lot of these compounds also modulate mitochondrial function and can uncouple, and they control calcium flows and all sorts of things. And so if we take on board Mike Levine's ideas, of course, they could potentially help reform or structure in the sense of structure through a boundary layer of magnetic fields, electromagnetic fields. And of course, this could also bring on board biophotons. Next slide, please. And I'll just finish with this one. One way of looking at life, which I thought might bring it all together, of course, is that life seems to accelerate entropy throughout the universe. And in this diagram on the y-axis, I'm showing time and entropy of the universe, and the x-axis is negative entropy. And you can see that if we follow all the things we discussed earlier, that quantum mechanics and thermodynamics seem to provide the laws which are necessary to create fields which organize chemicals, which brings life. And then the fields generated by life have a feedback mechanism. So you can see that all these things seem to integrate together. And the character on the right there, I suppose, some of you may recognize him. It's Death from the Terry Pratchett series of Discworld. But of course, you know, entropy is all about equilibrium. So I'll end there. Thank you very much.
Alistair, thank you very much, and well done indeed for keeping to time. That's very, very, very helpful indeed. Philip, are you ready to present your slides?
I am.
Great, lovely. Over to you.
Yep. Let me see. Is that visible to everyone? Okay, great. And can you see my cursor? Okay, fantastic. So in my talk in March, I posed it with "What is life today?" hearkening back to Schrodinger's famous 1944 tract in which he deduced the aperiodicity of DNA from fundamental physical reasoning about the need that living systems have for an information storage molecule, right? But then I went on to describe how light is essential as a scaffold for organizing whole biology, whether at the cellular scale, or below, moving up to organs like the brain, which Li Hui Sai and even Masashi Iono talked about with their beautiful plasmodium models. And then I dreamed a little bit talking about how quantum theory, specifically the unitary deterministic evolution of the wave function combined with state reduction processes, makes us question this whole idea of what it means to be mortal, right? These life-death transition events. And so I'm going to summarize briefly what some of what I discussed in that talk. So for those who are unfamiliar, Schrodinger posed this idea of the paradox of Schrodinger's cat. And the idea is simple if you haven't heard it, which is you have a cat in a box combined with a quantum triggering event like a radioactive decay of an isotope. And there is some quantum probability that this isotope will decay, and there's some probability that it won't. And the decay is associated in the classical Schrodinger paradox of triggering a hammer that then hits a bottle of cyanide and then kills the cat, right? Because the poison leaks out and kills the cat. So this idea of a quantum event that then enables a macroscopic classical event is something that we are very familiar with. I think Alistair and others, even in their own papers, have shown that on the biological side, what happens when something like cyanide were to affect an organism. But in the case of this cat, and one of the unique features of quantum mechanics is this linearity, right? So what does linearity mean? It's this superposition of so-called macroscopic states, the idea of a live cat and a dead cat. And this superposition that you see here above, written in this kind of simple mathematical way, is something that enables us to think about unitary evolution of both the dead part of the wave function and the live part. But when we observe, of course, we have some type of state reduction that is non-deterministic. We see a dead cat or we see a live cat, right? So in a many worlds interpretation, which goes out of the sort of canonical Copenhagen interpretation, if you have a number of these binary quantum transition events where you either have a decay or you don't, you have in one unique world, as this number of these triggers increases and goes to infinity, you produce a so-called, what I define as a quantum immortal state, a surviving cat state that essentially never dies. Now, the obvious and maybe more difficult question is, for all the branches in which the cat dies, we assume that the life-death transition event is irreversible, right? Now, what happens to the body of the cat immediately after death? How dead is it? Like, do we say that, oh, it's 99.9% alive? Is it 90% alive? These are not trivial questions about what is a measure of life, right? Because this goes back to our definition of, Jacques Monod's teleological purpose to others who have very broad definitions of life in terms of pure information rather than materialistic atoms, of the cat, which are, post the life-death transition are almost identical. So what is the key ingredient in the life-death transition? So this obviously raises questions of eternal life in another universe or realm or space, Hilbert space, if you will. So I call this slide the future of cat biophotonics, but it's really the future of organismal biophotonics or life biophotonics. So take for a moment that we consider all biophotonic interactions in an organism, we encapsulate it in a state psi prime. So, but particularly the photonic interactions, right? So psi prime, this sum of the biophotonic interactions of the material cat, may give a direct readout of the life-death transition. And this is no philosopher armchair speculation, right? NASA, when they send probes to the moons of Jupiter and Saturn, this is a real consideration. I mean, life on Earth, Earth has been in the making 4.5 billion years. But when you go to Titan or Enceladus and you've got seas of ethane and methane, and the question is not, oh, does life exist? But it's like, how far along is that world to the development of life, right? And are there ways that we can measure sort of the life-death transition and understand more clearly what are the prerequisites to the formation of an informatic context in which chemistry can evolve to produce life? So it was recently shown in 2020 that you can actually even take the probabilistic state reduction, the measurement problem, which is the fundamental conundrum of quantum mechanics, and you can actually couple that, what was previously thought to be irreversible, you can weakly couple that non-deterministic state reduction to a deterministic system and make it reversible. So it was shown by a group at Yale that you can actually couple this to a deterministic system and then actually predict and reverse the quantum jump. So is it possible? Is it something that we can dream of having all the biophotonic states of an individual organism combined and then couple it to a deterministic system that can then control that life-death transition, guiding from one side of life to death and then back again? So keep in mind that psi prime is an entangled electromagnetic image of a material cat. So whereas a purely atomistic viewpoint would suggest, oh, life and death, there's a chemical signature, which in fact there are, right? Rigor mortis and other things that happen later on. But prior to those things, there are photonic signatures. And these are some things that, Jimmy Bell and others are working very diligently to investigate. So this raises questions. This idea of us being beings of light is no mere spiritual speculation, right? This redox biology, the idea that we've got these ion channel flows that are producing electromagnetic fields, there may be a possibility one day for really identifying ourselves in that way. So I just conclude with two quotes that I spoke from my talk in March. One from Freeman Dyson, late 70s, who really started this very precise or mathematical understanding of physics and biology, or even the precursor to the anthropic principle, the cosmological anthropic principle. But he said that life and intelligence were inextricable from the long-range future of the universe. And here's a very simple example of that, right? In stellar furnaces, we produce carbon, the heavier elements, and even plutonium. Earth, for 4.5 billion years, has had some of that plutonium from the stellar furnaces, right? But most of it has decayed, considering that the half-life of plutonium is about 80 million years. So most of it has all decayed naturally in the Earth's crust. However, in the 20th century, nuclear physicists started producing plutonium, right? And so without life and intelligence, we would never have had the existence of plutonium on a 4.5 billion year old Earth. This is remarkable, and it changes the trajectory of what possibilities are there for Earth in the cosmos, right? And this can be extended further on the cosmological scale, right? Tens of billions of years, right? So the last thing I concluded my talk with was discussing this idea of, if there is such a close relationship between the atoms of life and the information of light, right? Other thinkers have thought about this problem of the etherealization of consciousness. Can we imagine an organism that transcends the atoms, that is purely the consciousness or the replica of that consciousness informatically, such that it can be contained within some type of radiative field? And J.D. Bernal quotes this very well. But lest you be afraid about us losing our very familiar embodied consciousness, I did show in my talk a very simple back of the envelope calculation, which showed that human consciousness in particular exceeds by at least 16 orders of magnitude the most advanced exascale computing systems in the world. So not only do you have, we are not just a simple system of biochemical reactions, we are a self-referential Gödelian system of reactions, but also that has an inner and an outer self, right? An internal world and an outer world that can then interface. But then also there is this idea that even if we were to replicate our consciousness, it would only be a limited version, at least with the present computational power that we have at our hands. So I'll conclude with that, and thank you very much for the discussion that is hoping to ensue.
Philip, thank you very much. Very, very interesting, and I'm sure that's going to prompt a lot of discussion. So at this point, Bethany is going to present some of Li Huisai's slides. Bethany, are you ready to present now?
Yes, thanks, Jeffrey.
Okay, over to you then.
Hello, everyone. I'm Bethany Adams. I'm the Gaia Foundation Science Communications person. As Jeffrey's already explained, Dr. Tsai unfortunately can't be here today, but she kindly supplied these slides, which I'm going to summarize much more briefly than the other talks because I'm not an expert. So Dr. Tsai is a neuroscientist and the director of the Pikawa Institute for Learning and Memory in the Department for Brain and Cognitive Sciences at MIT. And she's well known for her work on neurological disorders that affect learning and memory, particularly Alzheimer's. The research of hers that I will be summarizing here is her recent, really exciting results that demonstrate the use of non-invasive sensory stimulation to treat Alzheimer's disease, and more generally to induce gamma entrainment and neuroprotection. Next slide, please. So I'm not sure how many of you have ever had an EEG, but I'm sure most of you are familiar with this concept of brain rhythms. Brain rhythms range from the very slow, so less than 1 hertz, to faster rhythms of hundreds of hertz. And the specific frequency that we're interested in today is the band from 30 to 80 hertz, which is conventionally known as gamma rhythms. While brain rhythms aren't completely understood yet, it seems that gamma rhythms appear to be involved in higher order cognition, such as attention or navigation and sensory processing. And neurological diseases also have an effect on brain rhythms. So multiple models of Alzheimer's disease in mice show reduced gamma rhythms. Next slide, please. What Dr. Tsai's recent research demonstrates is a link between gamma rhythms, the amyloid clamping prevalent in Alzheimer's disease, and the use of 40 hertz flickering light stimulation. So flickering light at 40 hertz, which is in this gamma band of brain rhythms, it increased gamma rhythm power and reduced the amyloid protein clamping, which you can see represented in those two brain scans with constant light and then the gamma entrainment with flickering light at 40 hertz, which is quite a remarkable, the red dots are the amyloid plaque, and it's quite a remarkable reduction in the, as opposed to the constant light. Next slide, please. So as you might have noticed from the last slide, Dr. Tsai has named this treatment GENUS, or gamma entrainment using sensory stimuli. And given that vision is not our only sense, her next step was to look at other senses. Specifically, she started with hearing. Next slide, please. What I think is completely amazing in this research is that combining visual and auditory stimulation at 40 hertz increased this neuroprotective effect and involved other regions of the brain. And it reduced both amyloid and tau pathology. It counted brain inflammation and prevented neuron death across brain regions. Then in behavioral studies, it was shown to restore cognitive function. The suggested mechanism by which this treatment works seems to be that it elicits a response in brain microglia and astrocytes, which play a role in cleaning up brain debris. It also had an effect on brain vasculature, improving blood flow, as well as affecting neurons themselves. It's currently undergoing human trials across a range of subjects, including those with Alzheimer's disease. And so far, the data suggests that this treatment can reduce the loss of volume seen in Alzheimer's disease, as well as improve connectivity. That's the end of the slides.
Bethany, thank you very much indeed for presenting those slides. Let's move on now to Masashi. I think Masashi, you're going to show your own slides, are you?
Okay. So hello everyone. So I have introduced my experimental system, which combines a biological organism with some light stimulating system, so that this organism can search for solutions to computationally demanding problem. I started from a brief introduction of this organism. The name is Plasmodium of Trellis-Reimond Physarum polycephalum, which is a single-celled amoeboid organism. And you see this is a scale bar, so it's a quite large organism, but this is a single cell. And it's interesting that because there are many cell nuclei in this body, once you cut the piece of the organism, this piece can survive as another individual, and more than one individual contact together, they're going to fuse into one single individual. And this organism has some life cycle, and the stage we are watching, this yellow state, is called plasmodium. And in this stage, this organism is kind of photoresponsive. Actually, this organism doesn't like light, so if you illuminate the part, this organism withdraws from the light. We created a kind of feedback system which measure the shape of this organism and illuminate the light at the some of the lanes in this system. So basically, this red and blue pixel represent that this organism has some oscillatory properties, and the red and blue pixel represent that the thickness increased or decreased respectively. And basically, this organism wants to maximize its body area so that it can maximize the nutrient absorption. But like I said, this organism doesn't like light. So once some of the lanes are illuminated, this organism tries to withdraw from the light. And you see this organism perform kind of spatial temporal oscillatory behavior. It searches for the shape which can minimize the risk of being illuminated by the light. And by doing this, this organism make a kind of trial and error and find one of feasible solution of the traveling salesman problem. Actually, this system has 64 lanes, each of which is labeled with the city name and its visiting order. So you will see that after several tens of minutes, this organism find some of the branches which can elongate in non-illuminated lanes. For example, so this one, C1 stands for that the salesman visited the city C first, H2 means the H second, D third, E fourth. And yeah, so these the combination of these 8 branches represent this kind of traveling tour, which has the length 128. But the purpose of this problem is to find the shortest circular route. And in this case, actually, this organism didn't get to the shortest route, which is the length 100. But in this worst case, the length was 200. So the one found by this organism was not the shortest, but a pretty good one. And this system, which we call bounce back control system, uses the light. And this is a very simple control principle where when the light is on, the branch should shrink. And when the light is off, the branch should expand. But only by doing this, following this control principle, the system goes into kind of local minimum, which means that the amoeba finds the route which has kind of longer route length. But because of this oscillatory phenomena, sometimes this branch expands even in the illuminated lane, and sometimes it shrinks even under non-illuminated lane with some probability. And because of these fluctuations, this organism can overcome this barrier and find a better solution. And I don't I won't go into the detail today, but I showed you that the behavior of these branches are temporary and spatially correlated. So this fluctuation of this organism is not purely random. So it's kind of oscillative, spatial temporal oscillative dynamics, which has some temporal and spatial correlations. And we showed that when this correlation is stronger, this organism tends to find a better solution, meaning that the length of the route found was shorter. And then, and also I showed you that this system can be somehow imitated by classical mechanics. So this circuit is purely classical. So we created this analog electric circuit, which can mimic the behavior of the amoeba's shrinking and expanding dynamics. And also this part calculates a kind of neural network dynamics with some product sum operations. So basically the similar behavior can be reproduced using classical mechanics. But what is interesting was that this is other study. We found that there's microtubules in this amoeba, and in this experiment, when the microtubules was disrupted by some reagents, microtubule disrupting reagents, the ability of this organism to make a decision to get to a chemical kind of food. So when this organism has certain microtubule networks, this organism can approach to the chemical attractant source. But when these networks of microtubules were disrupted, it loses this ability. And so, so this kind of microscopic structure have something to do with a macroscopic decision-making ability. But what we saw in our movie, the experiment, we used the other stage of the organism, which is called plasmodium. That is quite large. It's kind of millimeter or centimeter scale. But experiments doing for this microtubule disrupting reagents is kind of microscopic scale. So we should be careful about comparing these two different spatial scales. But anyway, so in my talk, I showed you some results that our amoeba-based problem solver has pretty good ability to find a good solution. And so there is some relationship between the light photosensitive activity and problem solving ability. And this is my own view, but I'm happy if we can discuss with this idea. But because our system is using some photo light stimulation, and that kind of light stimulation is affected by microtubules, and the lacking the microtubules makes the system loses the ability of the problem solving. So maybe we can connect microscopic scale and macroscopic scale, which means some intelligent or problem solving abilities in this same experimental platform. Yeah, that's it. That's all for my talk. Thank you.
Sashi, thank you very much indeed.
Okay.
Absolutely fascinating. And I think your final point will be a point for discussion afterwards. So that takes us now on to Michael. Michael, are you going to show your own? Yes, you're going to show your own slides. Off you go, Michael.
Thank you very much. So I'm just going to present a very brief recap of my talk, which focus on the selected chapters of how cells respond to and generate electromagnetic fields. Now, I use the title cells, but it would be more appropriate is to say how actually biomolecular components of cells can respond to and generate electromagnetic fields. So let me switch on the laser pointer. So the first part was on how external electromagnetic field can affect or interact with biomolecular systems in general. So what we are fostering is the perspective of trying to understand this question from bottom up by using molecular simulations and how these can inform us about interaction of electromagnetic field with with biomolecular systems. And this is actually a very nice method how to calibrate the molecular simulation parameters to experiment, because these directly, in this case of simple amino acid solutions, we can make predictions about measurable microwave properties of this biological system. So to that end, to get the experimental data, we also develop a technology which enables us to measure the electromagnetic properties from very small volumes. Particularly, we focus on a microwave band of a spectrum, and for that band we develop planar chips in microstructured chips, where we can use only a few micro to nanoliters to actually measure the complex permittivity of the sample. So this is something very important, because then we can work actually with the precious samples, for example, as microtubules. For example, here we could demonstrate that observing complex permittivity of the tubulin solution we can distinguish if by microwaves if the tubulin is self-assembled to microtubules or not. So this has also kinds of sensing uses. The other area which analyzes how electromagnetic field interacts with biomatter, it's more related to the biomatter perspective. So we do carry out at first, to guide us, molecular simulations where we apply electric component of electromagnetic field to the simulation, and then we watch what happens to the biomolecules. We always do, of course, include the water and ions to simulate the physiological conditions. I'm just not showing them here. So for example, we could show that the tubulin subunit of microtubules is influenced by the electric field by aligning the dipole moment with the electric field and also changing its secondary structure. And also we could demonstrate that the microtubule lattice is actually opened up by electric field. This is all molecular simulations, and we were eager to see if this is actually happening in reality, but it's something where we can really be the masters of the whole systems, because we see the motions of individual atoms in these systems. So again, to verify these molecular simulation predictions, we develop chips, where the idea is that we develop chips which enables us to deliver the electromagnetic field in the particular area of the sample. And these chips at the same time are compatible with advanced super resolution microscopies. So we actually can see what happens to the biosystems under study when being exposed to electromagnetic field live, so to say. So for example, we developed a system for a structured illumination microscope, where the chip was aligned with the objective, and also we had a whole assembly engineered. And what we could demonstrate that applying very short nanosecond electric pulses, we could completely remodel the cell cytoskeleton. What you can see here in fluorescence is labeled are the tips of microtubules. So actually we kind of can modulate the cell biology using very short electric pulses, very intense electric pulses too. So the second area which I covered in a larger extended talk was related to how electromagnetic field, now speaking in broad spectrum, can be generated by biological systems. So there, I covered in talk our hypothesis, which became quite popular, about capability of microtubules to vibrate at their particular eigen modes, or the intrinsic vibration modes. And now, considering that the microtubules are very charged, electrically charged, these bring us to actually to theoretical finding and prediction that microtubules should be generating electromechanical waves which can propagate along the microtubules and hypothetically serve as a kind of communication mechanisms from the cell periphery to the cell center. We also analyzed, given the size of the microtubules, what could be the vibrational electromagnetic spectrum of the dividing cell in one of our papers. The largest area of the talk, I focus on biological autoluminescence, which is also called as ultraviolet photon emission or biophoton emission, which is a fascinating phenomenon of basically describing the fact that all biological systems do emit a small amount of light, which is non-thermal light. And so far, we know that the strongest evidence that this light comes from the energy for this light comes from the chemical reactions, where in the beginning there is typically a biomolecule reacting with the oxidant, which then in chain of reactions generates electronically excited state of a molecule, which with some probability can decay to generate a photon. So this is interesting phenomenon because, well, we can potentially use it for diagnostic purposes. For example, we could distinguish the types of the cells by measuring their spectra. We can also image this phenomenon and to see the unseen, because there is probably no other method which can label-free detect the areas which undergo increased oxidative stress. So for example, in this case, you can see the black and white hand of the subject. There's a black and white image, and here you can see the endogenous light signals coming from the skin and the free spots which you couldn't see otherwise by other methods, which where we applied hydrogen peroxide and basically caused oxidative stress on the skin. Furthermore, we also covered there are interesting, potentially interesting properties in the signals of these biological luminescence. Basically, we asked the question if there is any hidden information in the way how the photons come after each other in time. And there is a lot of controversy and interesting topics in this area. But I think there might be some areas related to the Hurst exponent properties, which might be giving some interesting information about these signals. So this is it in short, and thank you very much. I'll be glad to discuss any of these in more detail.
Michael, thank you very much indeed. Very, very concise. And that leads us to our final summarial talk, which is from Judith. Judith, are you ready with your slides?
Yes. Can I use you're showing the slides for me. Can I get a laser pointer activated or not? I guess not.
You'd have to share your own slides, Judith, to do that.
All right. Well, we won't waste the time. I think. Can you see you can't see a cursor, I suppose.
Yeah.
Can you see anything? No, not from my end. Okay, we'll just go quickly. What I wanted to address in my talk was, where are the data that truly implicate quantum mechanics in life? I think this is something that many people have been trying to demonstrate. And as a result of our work and others' earlier work on the role of electron transfer, I think there's now a strong body of evidence that nuclei, hydrogen, tunnels under biological conditions. So this idea of a particle moving through a barrier rather than over the top of the barrier. And this is going to apply in the very large number of redox reactions that involve transfer of electrons and hydrogen in biological interconversions, generation of energy, ATP, etc. Next slide, please. I wanted to point out how a very simple observation can lead to the development of a field. And that has to do with the properties of tunneling near room temperature. And I, on the left, highlight a figure from a book by Bell, published a long time ago in 1980, where he looked at the temperature dependence of the transfer of hydrogen as over a wide range of temperatures, and showed that in the semi-classical regime, which occurs as we raise the temperature, there's less and less quantum behavior. And the quantum behavior really only shows up, the extreme quantum behavior only shows up as you go near absolute zero. And that's where these hydrogens or the isotopic analog deuterium start to tunnel through the barrier. And the observation was made that there's no difference in energy for the tunneling of either protium or deuterium. The slopes are basically the same. Now, when we go to biological systems, we're looking at a very different time regime. And the unusual observation is that the slopes of the lines for protium and deuterium are still the same. But here, of course, biological reactions are temperature dependent. And so there's a positive slope which doesn't change on isotopic substitution. And this may sound like a simple deviation, but it's at the core, I think, of our understanding of how the large enzymes, which are the workhorse of the cell, can use their scaffolds to enable the movement of small particles via quantum mechanics. Next slide. Now, this is a summary of really decades of work, and I won't elaborate it in this brief summary of my longer talk. But essentially, it's the multidimensional quality of both electron and nuclear tunneling. And the difference with nuclear tunneling is, of course, the larger particle, the smaller wavelength. How do we accommodate that, as well as the ability to interrogate isotopic variants of hydrogen, such as deuterium and tritium. And to just cut to the chase, the way we think about this is that the large scaffold of the protein is moving all the time, it's in motion all the time, and moving from one conformational state to another until it reaches this transient degeneracy between the reactant and product wells, which is the place in which the particle can act as a wave and delocalize between the reactant and product wells through wave function overlap. These are temperature-dependent processes. However, the movement of the wave is temperature independent, but isotope dependent. We've separated out the thermal activation of the process from the inherent quantum wave particle duality. And that has to lie within the protein scaffold. The protein scaffold is basically classical. And so next slide, please. What we have done is spent the last 5 years trying to develop tools that allow us to look at the spatial resolution of this thermal activation as a starting point for understanding how the protein scaffold enables active site quantum behavior. And this is a result of one of the systems that I went into depth during my full presentation. And what I want you to see is that red arrow. Essentially, we were able, using this tool called temperature-dependent hydrogen deuterium exchange by mass spec, to show a conduit from the interface of the water or the solvent and the protein up to 20 angstroms away into the active site. So this is a thermal conduit for the creation of a state within the active site, transient creation within the active site that then, as I said, enables the wave function overlap. Next slide, please. Now, that's the spatial part of it. What about the temporal resolution? And we have been similarly trying to develop tools such as Stokes shifts, where we could look at nanosecond protein motions that, in fact, in recent data indicate if we put a probe on the exterior of the protein and look at the thermal activation and its energy of activation, and then compare that to the chemistry that's happening where that iron is quite far away, we find identical energies of activation, whether we're looking at something out on the protein solvent surface or buried in the active site. So this has led to the idea of protein quakes. Now, protein quakes are not new to biochemistry, but they've always been thought of as deriving from, for example, photoexcitation in the interior of a protein, which then gives heat dissipation out into the solvent. We're turning this around and saying that there is a thermal quake that initiates at the solvent interface and moves inward as the origin of the thermal activation and catalysis. Now, this idea of site-specific protein quakes is quite new experimentally, but has been in the literature as a theoretical premise for quite a while. And we have, I think, the tools and now the data to implicate these far from equilibrium. These are not equilibrated processes. These are out of equilibrium protein quakes. Next slide, please. Okay, let's just sum up. We have extended the temperature-dependent HDX mass spec to about 5 enzymes. These are very involved and somewhat tedious methods. We're trying to use computation to predict these site-specific thermal conduits as well. But let me just say, On the top, you see two well-demonstrated hydrogen tunneling systems. The thermal conduits are unique in each enzyme. They are specific to the reaction that's being catalyzed. If we move into the movement of a methyl group, much larger particle, unlikely to be transferred via a tunneling process, nonetheless, we still see a reaction site-specific thermal conduit distinct from, as you can see, the two on the upper half of the slide. Next slide, please. And then if we move into one of the largest scaffolds in biology for proteins, the Tim-Barrow enzymes, we've interrogated two so far. The top one does a deamination reaction. It's a monomeric protein. The bottom one is a dimeric protein. Involves the generation of PEP through the elimination of water from its precursor. These enzymes have different oligomeric structures, but very common scaffolds we find within the interior of the catalytic face of these scaffolds, totally different thermal networks depending on the reaction being catalyzed. So here is our premise. Our premise is that in evolution, we're not only attempting within the active site to get everything right, to get the actual alignments and components that give us these enormous rate accelerations, but that there is co-evolution of the protein scaffold along with the chemistry of the active site. And the concluding slide, please. The evidence for hydrogen, as well as, of course, electron tunneling, is a widespread component of biological reactions, I think is in hand right now. And we have analytical models that can describe the experimental observations. Now, the new thing that has come out is this combination of site-specific regions of proteins that allow for what we are proposing to be protein quakes that originate from the protein solvent interface and transiently enable these quantum mechanical wave function overlaps. So I think, for us, we really are basic scientists. There's so many unanswered questions. This remains a controversial area. And we are currently categorizing all of the protein motifs that appear to enable these protein quakes. And we really need a hardcore mechanism for site-specific heat transfer in protein. But what I want to point out is this question of life at equilibrium versus out of equilibrium, because in this very reductionist view of how proteins work, there are certainly equilibrated protein conformational substates, and this dominates the analysis of protein dynamics in the literature. But what we think has to be added on are these non-equilibrated, out of equilibrium protein quakes that are going to play a role not only in catalysis, but in the regulation of enzyme function. If we think about the future, actually, once we're able, which we have not been successful with thus far, is to define the physical principles that underlie enzyme catalysis, I think the field becomes wide open in providing templates for de novo catalyst design. And with that, I'll finish. Thank you.
Judith, thank you so much indeed, and well done indeed to all the speakers to have kept so well to time. I think that that session of refreshing what we've heard about during the spring series has been very helpful.